स्टूडेंट्स, जिवें कि तुसी जाणदे हो कि मनुखी शरीर विच किडनी दा एक मुख काम शरीर दे लहू विचों व्यर्थ पदार्थां नूं यूरिन दी फॉर्म विच बाहर कढना है। यूरिन राही शरीर तो विषैले ਅਤੇ व्यर्थ पदार्थ ਤਾਂ ਬਾਹਰ ਆਉਂਦੇ ਹੀ ਹਨ, ਪਰ ਤੁਸੀਂ ਇਹ ਜਾਣ ਕੇ ਹੈਰਾਨ ਹੋਵੋਗੇ ਕਿ ਇਹ ਮਨੁੱਖੀ ਸਿਹਤ ਦਾ ਇੱਕ ਮਹੱਤਵਪੂਰਨ ਸੂਚਕ ਵੀ ਹੈ। ਇਸ ਦਾ ਰੰਗ, ਇਸ ਦੀ ਮਾਤਰਾ, ਇਸ ਦੀ ਗੰਧ ਆਦਿ ਸਰੀਰ ਦੇ ਅੰਦਰੂਨੀ ਹਾਲਾਤਾਂ ਦਾ ਵੇਰਵਾ ਦਿੰਦੇ ਹਨ। ਯੂਰਿਨ ਦਾ ਰੰਗ, pH ਲੈਵਲ, ਗਾੜਾਪਨ ਆਦਿ ਸਰੀਰ ਦੀ ਕਿਸੇ ਅੰਦਰੂਨੀ ਕਾਰਜ ਪ੍ਰਣਾਲੀ ਵਿੱਚ ਗੜਬੜੀ ਦਾ ਸੰਕੇਤ ਦਿੰਦਾ ਹੈ। ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਅਸੀਂ ਯੂਰਿਨ ਅਤੇ ਮਨੁੱਖੀ ਸਿਹਤ ਵਿਚਲੇ ਸਹੀ ਸੰਬੰਧ ਨੂੰ ਜਾਂਚਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰਾਂਗੇ। तो चलो शुरू करते हैं। Let us start with general characteristics of urine. Volume. Normally 1.5 to 2 liter of urine is passed per day. Quantity of urine passed is less in summers as compared to winters. It also depends on the climatic conditions and intake of fluids by an individual. Color. It is usually amber yellow due to the presence of urochrome. This coloration is due to end product of hemoglobin breakdown. As far as transparency is concerned, urine of a healthy person is usually clear and transparent. Odor. It is usually aromatic when fresh, but later turn ammoniacal due to degradation of urea by bacteria. Specific gravity. The range of specific gravity of urine is 1.003 to 1.035 grams per cubic centimeter, which is slightly more than plasma. The pH of urine is close to neutral, which means pH of urine remain near 7 but can vary between 4.6 to 8. Normal constituents of urine. Normally, urine is composed of 95 to 96% of water and 4 to 5% solids. Solids include urea about 2% and some other nitrogenous substances like uric acid, creatine and creatinine. Organic substances like oxalic acid, lactic acid, amino acids and enzymes etc. are also present along with some inorganic salts. Abnormal constituents of urine. If we talk about abnormal constituents of urine, it is sugar in a diabetic patient, and in a kidney patient, it is albumin, a kind of protein commonly found by pathological tests. Bile is another abnormal constituent which can be detected in the urine of a jaundice patient. Glucose is the type of sugar found in blood. Normally, there is very little or no glucose in urine. In a serious diabetic patient, the blood sugar level is so high that its high volume can easily be detected in the urine. Glucose can also be found in urine when the kidneys are damaged or diseased. You all know that a large amount of waste products are produced during various metabolic activities in the body. Most of the waste are removed through the excretory system, but several other body systems also help in removing waste products from the body. The main contributor to excretion is the urinary system. Nephrons present in the kidneys filter the blood to remove waste products in the form of urine. Along with the other constituents we discussed, urea is the main nitrogenous waste of urine produced in the human body. apart from urea, urine contains ammonia, hormones, dead blood cells, proteins, salts and minerals and toxins. For many medical problems, urine tests are preferred over most other medical tests because they are extremely easy for the patients and they tend to reveal important information associated with the functioning of the body. Let us start with the test for the presence of sugar in a urine sample.
Objective. The objective of this experiment is to test the given sample of urine for the presence of sugar. Materials required. To perform this experiment, we need very ordinary lab materials like Benedict's solution, test tube, dropper, burner, etc. Procedure. As we know, urea is the principal nitrogenous waste product produced by the human body. The person suffering from certain metabolic or renal disorders may excrete sugar in the urine. Suppose this is the urine sample of such a person. To test sugar in the urine sample, take 2 milliliter sample of urine in a test tube. To this urine sample, add 2 milliliter of Benedict's reagent. Boil this solution. Cool it down to bring it to room temperature. On cooling down to room temperature, reddish-brown precipitates are formed. Conclusion. Appearance of reddish-colored precipitates in the solution confirms the presence of a large amount of sugar in the given sample. Let us perform another test to verify the presence of sugar in urine. Fehling's test for sugar. Material required. To perform this test, we need urine sample, Fehling's solution A, Fehling's solution B, test tube, burner, etc. Procedure. Take 2 milliliter sample of the urine in a test tube. To this, add 2 milliliter of Fehling's solution A. Also add 2 milliliter of Fehling's solution B to this test tube. Boil this solution. Cool it down to bring it to room temperature. On cooling, reddish-brown precipitates are formed.
Conclusion. Appearance of reddish-brown precipitates in the solution confirms the presence of a large amount of sugar in the given sample. Result of this test will vary with the presence of different concentration of sugar in different samples. If only traces of sugar are present in urine sample, yellow-colored precipitates will appear in the test. If moderate amount of glucose is present in any sample, light brown color will appear. The presence of large amount of sugar will produce reddish brown color after performing Fehling's or Benedict's test. This was about the test for the presence of sugar in a urine sample. Now let us talk about urea. Urea is one of the most important waste products our kidneys excrete. But how is urea generated in our body? As the liver breaks down amino acids, it releases ammonia. The liver then quickly combines that ammonia with carbon dioxide to produce urea, which is the primary nitrogenous end product of metabolism in humans. The liver turns the ammonia into urea because urea is less toxic than ammonia and can be temporarily stored in the body. Children, let us now perform experiments to test the presence of urea in urine.
Objective. Objective of this experiment is to test the given sample of urine for the presence of urea in it. Material required to test urea in the given urine sample, we need sodium hypobromide, urine sample, dropper, test tube. Procedure. To test the presence of urea in a urine sample, take 2 milliliter of the sample in a test tube. To this, add 5 drops of sodium hypobromide. Brisk effervescence of nitrogen appear. Conclusion. Appearance of brisk effervescence of nitrogen indicates the presence of urea in the given sample. There is another test to confirm the presence of urea in the urine sample. Urease test. Material required. For this test, we need 1% acetic acid, sodium hypobromide, phenol red, urease tablet, urine sample, test tube. Procedure. To test for the presence of urea in a urine sample, take 5 milliliter of the sample in a test tube. Add 4 to 5 drops of phenol red to it. Then add sodium hypobromide drop by drop till the solution becomes light pink. Then add 1% acetic acid drop by drop until the pink color disappears. Shake it if required. Add half the urease tablet in powdered form to the sample and shake it well. The solution turns red. Conclusion. Appearance of red color in the solution confirms the presence of urea in the given sample.
Students, you know that when excess amount of urea is present in urine, the condition is known as uremia. Another important constituent of urine is uric acid. It comes from the breakdown of nucleotides. Uric acid is insoluble, and when there is too much uric acid in blood, it will form crystals that may collect in the joints and cause gout. Gout is a disease that results when crystals of uric acid start getting accumulated in joints. This condition occurs when excess of uric acid is present in the body tissues, and due to it, the patient faces recurring attacks of arthritis. Chronic gout can lead not only hard lumps of uric acid in and around the joints, but also damage kidneys. Now let us talk about the constituent that we are going to test for next. It is albumin. It is a protein that is found in the blood of humans. Normally, the kidneys filter the blood and form urine. Proteins are retained in the blood, so there is no extensive loss of protein into the urine. Although healthy people do pass small amounts of protein in the urine, their presence above a certain level suggests that there may be malfunctioning of the filter mechanism. A good amount of protein may be present in the urine because of acute inflammation or kidney stone, or may be a sign of kidney damage. The presence of albumin in urine is called albuminuria. Let us perform a test to detect the presence of albumin in a given sample of urine.
Objective. Objective of this experiment is to test a given sample of urine for the presence of albumin in it. Materials required. To test a given sample of urine for the presence of albumin, we require test tubes, dropper, robot solution, urine sample. Procedure. To test for the presence of albumin in the urine sample, take 5 ml of robot solution in a test tube. Tilt the tube and add the urine sample with the help of a dropper. A white ring is formed over robot solution as we add the urine sample. So what do we conclude from it? Conclusion. Appearance of white ring in the solution confirms the presence of albumin in the given sample. There is another test to confirm the presence of albumin in the urine sample. It is known as the sulfosalicylic acid test. For this test, we need 30% sulfosalicylic acid and the sample. Take 2 ml of urine sample in a test tube. Add equal volume of 30% sulfosalicylic acid to it. Heat the tube gently. A cloudy turbidity appears in the solution. Conclusion. Appearance of a whitish or cloudy turbidity confirms the presence of albumin in the given urine sample.
Let us talk about another urine constituent, bile salts. Bile is a chemical produced in the liver and stored in the gallbladder. It aids in the digestion of fats and helps in the elimination of toxins from the body. Bile passes out of the liver through the bile duct and is stored in the gallbladder. After a meal, it is released into the small intestine. When the bile duct becomes blocked, bile builds up in the liver and jaundice develops due to the increasing levels of bilirubin in the blood. Bile salts appearing in urine is an indication of certain diseases, especially jaundice, and their quantities are detected by chemical tests.
Objective. Objective of this experiment is to test the given sample of urine for the presence of bile salts in it. Material required. Bile salts can be tested in the given sample by using Smith's reagent, dropper, test tube, and a sample of urine. Procedure: to test bile salts in urine sample, take 1 milliliter of Smith's reagent in a test tube. With the help of a dropper, add 2 to 3 milliliter of urine sample in tilted test tube containing Smith's reagent. A green ring is formed at the junction of Smith's solution and urine sample. Conclusion: appearance of a green ring in the solution confirms the presence of bile salts in the given sample. There is another test to confirm the presence of bile salts in the urine sample, that is, Pattern-Raffos test. Material required to perform this test are sucrose, test tube, concentrated sulfuric acid or H2SO4. For this, take 2 milliliter of urine and little sucrose in a test tube. Pour 2 milliliter of concentrated sulfuric acid along the sides of the test tube. Red color appears. Conclusion: appearance of red color in the solution indicates the presence of bile salts in the given sample.
Hope I have made a successful attempt to demonstrate various tests to detect the presence of sugar, urea, albumin and bile salts in the given samples of urine. Let us check what you have learned through these experiments. This question answer session will also help you to face Viva confidently during your practical exams. First question: what are the normal constituents of human urine? Answer: normally, the urine is composed of 95 to 96% of water and 4 to 5% solids. Urea up to 2% and some other nitrogenous substances like uric acid, creatine, creatinine constitute the solid portion. Organic substances like oxalic acid, lactic acid, amino acids, enzymes, etc. and some inorganic salts are also present in traces. Next question: where is urea formed in our body? Answer: urea is formed in the liver. Next question: out of the constituents given here, which one is a normal constituent of human urine? Sugar, albumin, urea or bile? Answer: urea is a normal constituent of human urine. As the liver breaks down amino acids, it releases ammonia. The liver then quickly combines that ammonia with carbon dioxide to produce urea. Urea is the primary nitrogenous end product of metabolism in humans. The next question is: when sugar is excreted in urine, which disease does it indicate? Answer: when sugar is excreted in urine, it indicates diabetes mellitus. The next question is: what does the appearance of yellow color in the Fehling's test for sugar indicate? Answer: if yellow color appears after performing Fehling's test for sugar, it reveals that traces of sugar are present in the urine sample. My next question is, what is albuminuria? Answer. The presence of albumin in urine is called albuminuria. The next question is, what is uremia? Answer. When excess amount of urea is present in urine, the condition is known as uremia. The next question is, on adding Smith's reagent to a urine sample, what does the appearance of a green ring indicate? Answer. Appearance of green ring in the solution confirms the presence of bile salts in the given urine sample. Next question. What causes gout? Answer. Uric acid is one of the constituents of urine. When there is excess amount of uric acid in blood, it forms crystals that collects in the joints and causes gout. Next question. On adding urine sample in a test tube having Roberts solution, a white ring appears. What does it indicate? Answer. A white ring in the solution confirms the presence of albumin in the given sample. Hope I have made a successful attempt in demonstrating experiments to test different substances like sugar, urea, albumin and bile salts etc. present in urine. I hope you enjoyed the class today. See you next time with another interesting set of experiments. Have a good day.